for attending today's webinar, NextSeq 1000-2000 Longer Reads and Extended Informatics. Before we begin, we wanted to cover a few housekeeping items. At the bottom of your screen are multiple application widgets you can use. All of the widgets are resizable and movable, so feel free to move them around to get the most out of your desktop space. You can expand your slide area or maximize it to full screen by clicking on the arrows in the top right corner. If you have any questions during the webcast, you can submit them through the Q&A widget. For the best viewing experience, we recommend using a wired internet connection and closing out any programs or browser sessions running in the background that could cause issues. Webinars are bandwidth intensive, so closing any unnecessary browser tabs will help conserve your bandwidth. The webcast is being streamed through your computer, so there is no dial-in number. For the best audio quality, please make sure your computer speakers or headset are turned on and the volume is up so you can hear the presenters. Some networks cause slides to advance more slowly than others, so logging off your VPN is recommended. If your slides are behind, pushing F5 on your Windows keyboard or Command-R on a Mac will refresh the page. You can find additional answers to some common technical issues located in the Help widget at the bottom of your screen. I would now like to introduce our first speaker, Sam Hester, a Senior Product Marketing Manager at Illumina.
Hi everyone, and thank you for joining us today for this webinar on the NextSeq 1000 and 2000 systems, where we will cover some of the new product updates on our flagship benchtop platforms, enabling new longer read methodologies, as well as providing some information on how the onboard informatic capabilities of the system can benefit your lab. I'm Sam Hester, Product Marketing Manager covering our benchtop systems, and I'm joined here by my colleague Deepthi Shankar, Product Marketing Manager covering our informatics portfolio. I wanted to begin today by going over the agenda for the next hour. We'll start with an introduction to both the NextSeq 1000 and 2000 systems and what make these systems unique compared to previous Illumina sequencers. And then we'll cover our new 600 cycle technology that was introduced on the system late last year and how they've been most commonly used by early customers. And then Deepthi will go over the onboard DRAGEN secondary analysis capabilities of the system and how they could streamline your bioinformatics pipelines in your lab. Finally, we'll cap off the presentation by sharing some early information on our new XSleep SBS chemistry that will be enabled on the NextSeq 1000 and 2000 systems early next year. And then we'll reserve some time at the end for questions, so please feel free to throw those into the chat as we move along. Let's kick things off with an introduction to the NextSeq 1000 and 2000 systems. The NextSeq 1000 and 2000 systems are the latest sequencers released by Illumina and are the result of well over a decade of experience and innovation in genomics. We've listened to your feedback and redesigned the system from the ground up with 4 key tenets in mind. Flexibility, with the ability to run anything from panels and small genomes to trios of whole human genomes. Efficiency, where we've touched on every part of the workflow from reagent cartridge to the control software to onboard secondary analysis to make this one of the simplest sequencers to use yet. Cost efficacy, where the number of technological innovations have allowed us to offer better pricing per sample on a benchtop than we've ever been able to do before. And scalability, where the range of throughput bridges the gap between methods from traditional benchtop systems such as MySeq all the way up to production scale systems such as the NovaSeq, resulting in a true mid-throughput platform. To enable these tenets, there have been more than 75 new technological innovations that differentiate these systems with our past sequencers. Some of the most significant ones include blue-green color chemistry that enables a stronger signal on our new ultra-high density pattern flow cells. We've also improved the optical system to better enable detection. The NextSeq 1000 and 2000 are also our first systems to contain onboard secondary analysis in the form of our DRAGEN Bio-IT platform, which Deepthi will elaborate on further in this presentation. And finally, we've redesigned our sequencing reagents with an integrated cartridge-contained design that streamlines the workflow and eliminates the need for pre- and post-instrument wash cycles. A lot of what I just mentioned contributes greatly to the efficiency improvements on the NextSeq 1000 and 2000 systems to make them by far our easiest to use systems yet. First, these systems contain an onboard secondary analysis platform in the form of the DRAGEN Bio-IT platform, our fastest and most accurate analysis option yet, and we'll go on into detail on that later in this presentation. Second, both the size and the number of components involved in sequencing have been reduced into a single integrated cartridge that aids in storage and organization. But another key component of this integrated cartridge is that it includes all the fluidic systems required for SBS, keeping the instrument itself dry, meaning there's no longer a need for pre or post wash cycles or a possibility of run-to-run carryover. This also greatly simplifies instrument maintenance. And third, we've streamlined run setup options to be as flexible and intuitive as possible. Beyond the workflow improvements, here's a look at all of the current flow cell configurations available on the NextSeq 1000 and 2000. We now have 11 different flow cell configurations on these systems, with more to come, spanning from P1, which offers 100 million reads and up to 60 gigabases of data, to P2, enabling 400 million reads and 180 gigabases of data, and scaling up to P3 with minimum specs of 1.2 billion reads and 360 gigabases of data at 300 cycles. This flexibility of flow cell outputs is complemented by a range of read length choices from 50 cycles to 600 cycles on our latest flow cells, enabling everything from counting applications such as bulk RNA-seq and single cell, up through longer read applications that we'll dive into in a few slides. So this slide is showing a more in-depth look of this, but don't worry about getting too bogged down in the details. I just wanted to show how this range of flexibility provides multiple efficient options for different project choices, from smaller batches being ideal on P1 and scaling up to larger studies that are enabled by P3, all at efficient runtimes ranging from 48 hours at max output down to just 10 hours for our quickest mid-throughput run, providing great quality data at every configuration. Here we can examine how these new innovations affect both throughput and cost of these systems compared to our legacy platforms. The NextSeq 1000 system was designed to match the legacy NextSeq 550 system with 120 gigabases or 400 million reads of output, but enables a much lower cost per gigabase for the user and is available at a lower capital cost. The NextSeq 2000 system increases the output possible on a benchtop to 360 gigabases or 1.2 billion reads of data, lowering the cost per gigabase even more. The increased output and lowered costs on these systems enables labs to run more samples at a lower price, or to take on more ambitious methods like single-cell RNA sequencing, exomes, and much more. To help enable you to access these benefits, Illumina has options to upgrade your existing NextSeq 500 or 550 or MySeq to a new NextSeq 1000 or 2000. Reach out to us for more information if you are interested in scaling up your capabilities while keeping per sample costs down, particularly for longer read applications. Though sample price is important, selecting a new sequencer isn't all about just the costs. The NextSeq 1000 and 2000 systems offer top-of-the-line benefits in terms of workflow through on-market compatibility with library preparation methods, no need of wash cycles, easy loading of reagents with a small consumable footprint, and offering easy access to bioinformatic pipelines through onboard DRAGEN. Additionally, the system offers the largest range of flow cell configurations and choices of any of our benchtop sequencers, with more to come early next year through the integration of XLEAP SBS chemistry, and read length options from 50 cycles to a longer 2x300 run available now. Lastly, choosing NextSeq and Illumina means choosing quality through a robust field-tested solution backed up by thousands of publications in just the last 3 years. Over 1,700 NextSeq 1000 or 2000s have been installed since their inception, and Illumina offers decades of experience through our award-winning service and support, customer care, and tech support teams to make sure you're supported through your NGS journey. Now that we've taken some time to discuss the NextSeq 1000 and 2000 systems, we wanted to dive into our latest products on the system, including new longer read configurations and their most common uses so far. Just at the end of last year and available now, Illumina has launched 3 new flow cells enabled on both the NextSeq 1000 and 2000 systems. We have a new P1 100 cycle kit with a rapid 10-hour runtime. This kit is spec'd to provide 100 million reads, and we think it'll be perfect for rapid detection and smaller projects for pilot studies for transcriptomics or single cell. We have also, for the first time, brought longer 600 cycle reads to the NextSeq 1000 and 2000 systems on both the P1 and P2 flow cells. The P1 flow cell will provide 60 gigabases of output with much better data quality and much faster runtime than the older MiSeq v3 chemistry. The P2 flow cell is a higher output version, provides 300 million reads and 180 gigabases of output with a still sub 2-day runtime. In terms of methods, over the past 3 years, the NextSeq has been a workhorse for single-cell, spatial, RNA-seq, and exomes. But we really think these newer kits are going to greatly increase the utility of the system overall by providing access to applications and methods traditionally locked on the MiSeq due to read length requirements, including immune repertoire profiling and a number of metagenomic applications. I'm going to mainly focus on providing examples of these applications newer to the NextSeq 1000 and 2000 in the next section. Let's start with one of the most common methods of benchtop sequencing for microbiology, small whole genome sequencing of bacteria and viruses. To show how the use of the new 600 cycle kit can compare to traditional uses, I wanted to start with a study we performed comparing the P1 600 cycle flow cell specifically to the older MiSeq v3 flow cell, looking at 3 bacterial samples prepared with Illumina DNA prep and with varying GC content from 35 to 69%. This slide shows a summary of the average metrics of these runs. The P1 flow cells on the NextSeq provided much higher data quality of 88% of bases called greater than Q30 across the run, compared to about 72% on the MiSeq. The P1 flow cell also provided higher output, above 60 gigabases, compared to just 15 gigabases on the MiSeq. And it did so at a much faster runtime of about 34 hours compared to about 56 on the MiSeq. Diving in deeper, there are 2 other specific aspects of this study I want to highlight. The first is shown on this slide, which is essentially that across the varying GC content of these 3 bacterial species, again ranging from 35 to 69%, the data from NextSeq P1 600 cycles was highly concordant with what is found on the MiSeq v3 chemistry, the previous gold standard for longer NGS reads. And second, the improved data quality of the P1 flow cell in comparison to the MiSeq across the run, which you could see here in the figure on the left. Here, NextSeq P1 600 cycles is shown in red, and MiSeq v3 600 cycles is shown in blue. Those of you who've run the MiSeq before are aware of the drop in data quality generally seen at the very end of a longer 2x300 run, especially in the last 30 cycles or so, and especially in read 2. This was one of our most significant areas of focus in improving the results on P1 and P2. You could see that in comparison to the MiSeq, the Q-scores of the NextSeq P1 remain very high, above 80% for nearly the entire run. And towards the end of the run, the drop-off isn't nearly as severe. In fact, at the end of the run, where MiSeq Q-scores generally drop off very significantly, the NextSeq P1 kit remains high, averaging 72.1% in the last 30 cycles of read 2, providing useful data throughout the entire run. Whole genome sequencing of isolates isn't always what you need in microbiology, however, as many times you're instead trying to identify what genus or species compose a mixed sample. That's where common techniques like 16S come in. 16S metagenomic sequencing has been a staple for bacterial identification for years, but for a long time limited to just the MiSeq due to the length of the V3, V4 region of the genome generally amplified by requiring a 2x300 run. This meant that labs running 16S metagenomic identification at scale would often have multiple MiSeq systems running constantly due to limited capacity available on a single MiSeq system. Labs also faced the choice on the MiSeq of running fewer samples at more depth, or sacrificing depth and resolution to fit more samples on a flow cell to increase economics and turnaround time, enabling labs to no longer have to choose between depth, speed, or costs for longer read applications was one of the major reasons for bringing these new kits to the NextSeq. However, for such an established method such as 16S, one of the most important things to do is establish strong concordance with the existing workflow, Here's one of the first slides we've ever shown on these new 600 cycle kits to show just that. Strong data concordance in identification and representation of bacterial species on a 16S project run on both NextSeq P1 and MySeq V3. We wanted to test metagenomic applications in customer hands to ensure the same results are achievable in the field as we found internally. Zymo Research Corporation was one testing site that mainly focused on 16S metagenomic sequencing. Zymo ran a comparison study on one of their standard 16S workflows, looking at the V3-V4 region, prepared using the Zymo Quick 16S NGS library prep kit. They then sequenced the same sample set on both the MySeq V3 600 cycle kit with a 15% Phi-X spike-in, and on the NextSeq P1 600 cycle kit with a 30% Phi-X spike-in. Zymo's main goal was to compare what they could do with the new flow cell with the same budget, and the P1 flow cell and MySeq V3 have comparable run costs. And even while accounting for the higher Phi-X spike-in, P1 provides about 3.3 times the output of the MySeq. First, Zymo shared that across these runs there was very high concordance between data uncovered on the MySeq and the NextSeq 2000, which fits very well with the data I just showed previously that was internally generated. Second, and more significantly, they provided these two figures, showing that by being able to devote both more reads and higher quality reads per sample, for the same price on the NextSeq P1 flow cell compared to the MySeq, they were able to uncover about 3 times more unique sequences from their samples, and through that resolve about 3 times the amount of observed species in comparison. Showing an example of our claim that labs running 16S everywhere will be able to take advantage of the higher output and improved quality to generate real improvements in their results. Zymo felt much the same about these kits, saying, "We love the P1 600 cycle kit. We are so impressed by the pricing and data quality improvement. This kit supports our mission of providing the highest quality data to our customers, and the shorter sequencing time allows us to be more responsive to our customers with quick turnaround times." Transitioning away from metagenomic samples, immune repertoire profiling is another method that is seeing strong use in the first 6 months of these new longer read kits on the NextSeq. Immune repertoire profiling of the full VDJ region is an application that requires a full 600 cycles of sequencing or a 2x300 base pair run. And like 16S until now, this has been historically only possible on the MySeq V3 kit. This chart here shows why this is. You can see here different metrics from sequencing the VDJ region on a MySeq at different read lengths, including 2x250, 2x275, and 2x300. Due to the length of the region and the need for overlapped and aligned reads, anything shorter than 2x300 results in a loss of clonotypes detected. However, the lack of output and lower quality at the end of the read on a MySeq really limited the depth and scale of what you could accomplish with an immune repertoire study. This is precisely what we were trying to address by bringing these new flow cells to the NextSeq. On the new NextSeq P1 and P2 flow cells, we significantly increased output and quality across the read, especially at the end, as we've shown earlier. This chart here shows that precise quality improvement in effect. Under downsampled conditions, the new NextSeq kits provide a significantly lower percentage of reads dropping due to failed mapping, and therefore a higher overall clonotype count. Since the sequencing depth was the same on each system, it's mainly the quality of the read contributing to this improvement. If you extrapolate this over the ability to sequence more samples in the same flow cell, or a much greater depth per sample, you could see how these new kits are going to unlock greater versatility in clonotype detection for immune repertoire profiling. Here we're just showing that we tested multiple different conditions, including sequencing depth, input quantity, and even antibody type. and that higher detection of clonotypes on the new flow cells was consistent across all conditions. Something else to note on this slide, you can look from left to right and see a 2 to 3 times increase in the number of clonotypes detected as you move from 2 million reads to 10 million reads per sample. So greater depth does significantly affect the discovering power when analyzing the immunome. So having greater output alongside improved data quality on the new P1 and P2 flow cells should have a high impact in this area. Moving to another user example with user-generated data, this study was run by Alchemab Therapeutics, one of our KOL testing sites for immune repertoire profiling. We provided kits for Alchemab to perform a comparative study on how the NextSeq P1 and P2 flow cells would perform alongside the MySeq V3 kit that they've traditionally used. You could see here that Alchemab used a unique library structure along with a long VH region in the center and primers and a unique barcode and UMI per sample that would require a 2x300 run to resolve quality data. They tested these kits across libraries with various levels of estimated B cells from 250,000 up through 1 million and 2 million. What they found is that the P1 and P2 kits resulted in an increased number of raw reads, as well as improved sequencing metrics across all libraries, which they found especially impressive given that they characterized these libraries as some of the most difficult to sequence. This is to be expected, though, with the new flow cells, so let's look at what an improved number of quality reads allowed them to do. Alchemab found that when analyzing the counts per UMI within each library, the NextSeq flow cells provided a significantly higher consensus count, or more reads per UMI, than the MySeq. You can see in the bottom left a chart that shows the majority of UMIs on the MySeq, above 60%, had a consensus count of just 1, where around 50% of the UMIs on each of the NextSeq flow cells had a consensus count of 3 or more. To ensure quality results, Alchemab applied a filter of greater than 2 for UMI consensus counts to each library, and found that under these conditions, the NextSeq flow cells provided more unique sequences and better retrieval of libraries with greater quantities of B cells compared to the MySeq. You can see in the chart on the bottom right, this is especially true in libraries where the expected B cell count is 1 to 2 million. And the NextSeq P1 and P2 both had a greater count of unique sequences compared to the MySeq. On this slide, the figure on the left is another look at quantifying the unique sequences uncovered by all 3 flow cell types. It shows greater counts of unique sequences uncovered by NextSeq P1 and P2, but it also shows high concordance in sequences uncovered between the systems, meaning that P1 uncovered nearly all of the sequence that MySeq did, plus significantly more, and P2 uncovered nearly all that P1 did, plus even more than that. Alchemab reported that in their downstream analysis, these unique sequences contributed to uncovering more unique features of the repertoire. One example I've included here on the bottom right was their report of the HCDR3 length uncovered by all 3 kits. Due to the unique sequences uncovered, NextSeq P1 and P2 were able to provide insight to a longer HCDR3 region, which was of particular importance to them. Ultimately, Alchemab had a very positive impression of the new P1 and P2 kits, saying, the NextSeq showed a clear improvement over the MySeq in terms of both the quantity and quality of antibody sequence data produced. Using the NextSeq at Alchemab enables us to more deeply mine the antibody repertoire at reduced cost, allowing us to discover the very best protective antibodies, even if rare. We don't have time to go into all of the data, but we did get similar feedback from other KOL sites studying the immunome, including Takara Bio, who similarly reported that Illumina's new 600 cycle kit for the NextSeq 2000 will be a critical piece in future immune profiling studies, as researchers no longer have to choose between high data throughput and full-length VDJ information. And with that, I'll turn things over to Deepthi to talk about onboard DRAGEN.
Thank you, Sam. Hi, everyone. I'm Deepthi Shankar, product marketer at Illumina covering informatics. Sam, that was a fantastic overview of the longer cycle kits and the key application areas. Let us now look at another great benefit of NextSeq 1000-2000, which is onboard secondary analysis with DRAGEN. So what exactly is DRAGEN? DRAGEN, or Dynamic Read Analysis for Genomics, is a collection of hardware-accelerated bioinformatics pipelines used for secondary analysis. This happens right after sequencing. DRAGEN takes reads and creates variants or counts depending on the application. The output from DRAGEN feeds into tertiary analysis where genetic data is interpreted. DRAGEN has been designed to be our most accurate, comprehensive, and efficient secondary analysis solution to date. NextSeq 1000-2000 is one of the first systems to have DRAGEN secondary analysis on board. This is a paradigm shift for sequencers where you can get variants as a direct output from the sequencer. Let's look at the benefits for a minute. It's accessible and easy to use, allowing you to do secondary analysis on the instrument with an easy-to-use graphical interface. It's cost-efficient. Onboard DRAGEN hardware and license is included with the NextSeq 1000-2000 system, so you don't have to pay for analysis that you do on board the sequencer, and there are no additional hardware costs. It comes with DRAGEN original read-only compression, which reduces FASTQ file size, storage, and energy costs by up to 80%, all without loss of data. And it's highly accurate. It generates high-quality variants directly on board NextSeq 1000 and 2000 systems. NextSeq 1000-2000 comes with some of the most popular DRAGEN pipelines integrated on board. They include BCL convert, enrichment, DRAGEN RNA, DRAGEN germline, DRAGEN single-cell RNA, and the latest pipelines on board include DRAGEN Amplicon and DRAGEN COVID-seq applications. These pipelines account for more than 80% of all the DRAGEN analysis we see, and which matches the most commonly used applications we see on the system, such as exome sequencing and bulk and single-cell transcriptomics. In the NextSeq 1000-2000, you have a choice of run setup and analysis options with DRAGEN on board. It is important to us to give you options when choosing the best path for each individual workflow. Customers who prefer to run locally can upload sample sheets directly to the sequencer and now use onboard DRAGEN to complete everything in-house. Alternatively, you could choose to set up your run with intuitive run setup in BaseSpace and use DRAGEN analysis in BaseSpace to access pipelines not on board the NextSeq 1000 and 2000. And of course, you could mix and match with a hybrid approach to find the best solution for you. DRAGEN is renowned for its accuracy as demonstrated by the Precision FDA Truth Challenges. In 2020, DRAGEN won the most accurate secondary analysis in all benchmark regions, and with each subsequent release, we have demonstrated further accuracy improvements with the Precision FDA benchmarking data. Late last year, DRAGEN also won the Somatic Indel Challenge for best applicability and generalizability, meaning it can be more widely applied to novel panels with the highest accuracy. We continue to bring these advancements on board the instruments, and you can access the latest pipelines on cloud via BaseSpace Sequence Hub. One of the significant benefits of DRAGEN on board is the accessibility and ease of use. Multiple customers have claimed that DRAGEN on board NextSeq 1000-2000 has increased runs per week, from around 3 runs per week to about 5 runs per week. Here is an example project that is pretty common with our customers that use the P2 2x100 kit that provides 400 million reads to run 16 samples per batch. You can see that in their traditional work week, you'll need about 21-hour sequencing run, and then you pass it on to a team to do bioinformatics offline that validates run success with those libraries and do the analysis. This easily bleeds into 2 days, which limits the number of batches they can do per week. Now with DRAGEN on board, users can do a run per day. You walk in the next day, the run and analysis are complete. Not only are they able to do a run per day, but they are also able to validate and look at the run metrics during that workflow. This allows them to go from about 48 exomes to 80 exomes per week. That's almost 60% increase in throughput. We continue to bring updates to onboard DRAGEN. DRAGEN V310 can now be used to generate FASTQ files on board for the 600 cycle kits. It has improved memory allocation, enabling successful processing of longer read data. And as we just heard from Sam, these new longer read kits support 16S metagenomics, immune repertoire profiling, shotgun metagenomics, and more. Another key update with DRAGEN V310 is that you can now call copy number variants with enrichment application for NextSeq 1000-2000. The DRAGEN enrichment workflow can now be used to call small variants, structural variants, and copy number variants for germline and somatic samples. Efficiency and usability improvements have also been made to germline, amplicon, RNA, and single-cell apps. Our goal is to continue to bring new applications and enable more flexibility on NextSeq 1000-2000 systems. To that end, we continuously update DRAGEN onboard NextSeq 1000-2000. We doubled the number of applications, tripled the supported flow cell types, and there have been 3 version updates in the last 3 years with added support for single cell, spatial transcriptomics, infectious disease, and amplicon panels. Now let's go into one of the key application areas, enrichment. Exomes account for approximately 2% of the human genome, yet 85% of genetic mutations known to cause disease occur in the exomes. By using exome enrichment, you can isolate and separate relevant regions for focused analysis. There are several advantages to exome enrichment. It has a broad range of applications from genetic diseases to oncology and population genomics. It's faster and easier compared to whole genome analysis. It's cost-effective, yet provides comprehensive coverage of the coding regions and provides significant improvement in findings compared to small targeted panels. Let's look at an example of this workflow on both the NextSeq 1000 and 2000. Once you have your sample, you prepare your library using Illumina DNA Prep with Exome 2+ enrichment. Then you could run your sample on the P2-200 cycle kit on NextSeq 1000 or the P3-200 cycle kit on the NextSeq 2000. Finally, the DRAGEN enrichment on board or the DRAGEN enrichment app on BaseSpace Sequence Hub helps you with secondary analysis. You can then use the results for downstream interpretation. The onboard DRAGEN enrichment pipeline provides rapid analysis for increased accuracy from enrichment and hybridization-based exome panels. It can run in germline or somatic mode. BCL Convert provides FASTQ files, samples, and go through the mapping and aligning process, followed by precision sorting and variant calling. You can enable structural variant calling, and with the latest 310 update, onboard NextSeq 1000-2000, you can also enable copy number variant calling with panel of normals in both germline and somatic modes. Output files include VCF, GVCF, and or BAM files. Note that GVCF is generated only in germline mode, and multiple VCFs will be generated if copy number variant calling or structural variant calling are enabled. Doing a quick comparison with BWA-GATK, one of the most commonly used analysis pipelines, DRAGEN enrichment is faster and more accurate. It has higher precision, higher recall, and overall accuracy for SNV, and in particular for indels. The F1 score, which is the overall accuracy score, which is what you see here on the right side of the table, is 89.74 for DRAGEN versus 82.1 for BWA-GATK. And in addition to the accuracy, DRAGEN is also fast. For instance, a flow cell on NextSeq 1000-2000 can be processed in less than 2 hours. DRAGEN enrichment application on board NextSeq 1000-2000 is not only fast and accurate, it's also very easy to use with a one-step run setup and onboard analysis. You can easily set up the run using a clean graphical user interface directly on the instrument or on the cloud using Base Sequence Hub. And during this run setup process, for enrichment application, you can choose to enable CNV and import panel of normals. Easy graphical user interface brings DRAGEN to all users. With DRAGEN onboard NextSeq 1000-2000, you can easily track run and analysis progress along with data quality metrics. You get per sample plots for all workflows with DRAGEN FastQC, and for the enrichment workflow, you can get aggregated output as an HTML report, or multiple metrics per sample are available as CSV files. I would like to share a quote from one of our customers, Dr. Tonya Carter, who uses NextSeq 1000 for their research and points to the value of DRAGEN on board. DRAGEN onboard's fast and accurate generation of QC metrics, alignment files, and median call files has markedly streamlined our processing of sequencing read data. The efficiency of this workflow has reduced our sequencing turnaround time, allowing us to perform more sequencing runs per week. A distinct benefit of having DRAGEN on board integrated into our sequencer is that raw sequence data can be processed even if your knowledge of bioinformatics pipelines is limited. So to summarize here, you can maximize turnaround and minimize costs with DRAGEN onboard NextSeq 1000-2000. It's accessible and easy to use secondary analysis, it's cost efficient, and it's highly accurate, generating high-quality variant calls directly on board your sequencer. And with that, I'll pass it over to Sam. Thank you all.
Thanks, Deepthi. I wanted to wrap up today's presentation with a very brief look ahead at how our new XLEAP SBS chemistry will impact the NextSeq 1000 and 2000 portfolio. As you can see across 3 years of the NextSeq 1000 and 2000 systems, we have been continuously innovating new products and additions to improve user experience and options. The latest being the 3 new kits, including 2 new longer read flow cells that we spent time discussing today. But looking ahead, we aren't done. We've also announced that early next year, we're going to be bringing our new XLEAP SBS chemistry to the NextSeq 1000 and 2000, first in the form of a P4 flow cell, and eventually backwards compatibility with our other flow cell configurations. So there's plenty of innovation coming on these systems. On this slide, I'm incredibly excited to share some of the first data to come off XLEAP chemistry on board the NextSeq 1000 and 2000 systems specifically, which shows a 50% reduction in both phasing and pre-phasing on the system, opening the door for both improved quality and performance in the future. But more importantly, I wanted to leave you with a detailed description of everything we've announced will come with the XLEAP 2024 update. First, we plan to release a new P4 flow cell that will enable more than 500 gigabases of data with 1.7 billion reads, representing a 40% increase over current P3 levels and enabling the lowest cost per gigabase ever on an Illumina benchtop instrument. After this, the next wave of updates will revamp the existing configurations from P1 to P3, lowering pricing for each kit and delivering improved runtime and performance. The new XLEAP SBX kits will also enable cost-efficient integration with our future library prep portfolio, notably our Illumina Complete Long Read targeted kits coming later this year. Finally, it's our intention to make these improvements available for all NextSeq 1000 and 2000 owners without any sort of purchase or priced upgrade, so XLEAP will be enabled with a free software upgrade to the system upon release. To wrap up our session today, we'd like to take a moment to share some resources with you where you can learn more about NextSeq 1000 and 2000 and onboard DRAGEN. First, here are some QR codes that will lead you to the specification sheet for the NextSeq 1000 and 2000 and to the data sheet with more information about DRAGEN onboard specifically. Next, here are QR codes that will lead you to the resources for the methods discussed here today: small whole genome sequencing, 16S metagenomic sequencing, and immune repertoire profiling. And then here are QR codes that will lead you to resources for the 3 most common methods used on board our flagship systems: RNA sequencing, exome sequencing, and single-cell transcriptomics. And with that, thank you very much for joining us today. At this time, we'll open the floor to questions. So the first question is, can you go into more detail on the new 100 cycle kits mentioned and how you see those used? Absolutely can do that. I didn't want to touch too much on the new 100 cycle kits because between all of the major topics covered, I didn't want to branch too far out of scope, but we do have new lower output 100 cycle kits on the P1 flow cell that provide a minimum of 100 million reads. And this new kit has a runtime of about 10 hours. So we do see it being impactful for gathering rapid answers for microbial surveillance or really any method looking for sort of fast counting application answers. We've also seen a lot of early customers doing shallower RNA-seq projects or for pilot studies, a couple samples doing deeper transcriptomic projects or even single cell or spatial sequencing. Looks like the next question is, does the NextSeq platform perform as well in low diversity samples as the MiSeq? Definitely. We've tested and validated multiple lower diversity methods on the new NextSeq flow cells, and we're able to get great results, some of which we shared today. Adopting any new platform requires some adjustment and fine-tuning of things like loading concentration or Phi X spike-in for your method. But both we internally and a lot of our customers who've tried it so far have been able to get both highly concordant and better quality data than MiSeq. As you saw in one of the examples, NextSeq does require a little bit more Phi X than the MiSeq at times, but even with that higher level Phi X spike-in, I want to remind you that P1 still provides more than 3 times the output of the MiSeq kits with much better data quality for about the same cost per run. So if you're still getting a great deal even when considering that Phi X. And we do have some creative customers already who are finding solutions to not waste reads on Phi X, such as combining 16S samples on the same flow cell with a highly diverse library, such as shotgun metagenomic libraries. All right, what's the next question? Looks like the next one that we're getting popped up, sorry, there's a little bit of a delay here, is that most labs I know still run the NextSeq 500 or MiSeq. Why do you think they haven't switched to the NextSeq 2000 yet? That's a good question. I would say that the NextSeq 500, 550, as well as the MiSeq, those are all great instruments. They work really well, and they've been good lab partners for our customers for many years. Buying new sequencers isn't a decision that requires capital investment, and many labs run under the if it's not broke, sort of don't fix it philosophy, you know? So even with the more directly visible pricing, quality, output improvements on the NextSeq, those systems can still be sort of a tough decision to switch over. But I do think that NextSeq 550 provides a great frame of reference to the other aspects of the value on NextSeq 1000, 2000 provide beyond those things. Customers who've switched absolutely rave about the quality of life differences on the systems and how various aspects on the 1000 or the 2000 improve total cost of ownership. So like the lack of wash cycles, easing the workflow burden, or lack of downtime due to fluidics maintenance issues, or things like smaller reagent footprints in your freezer compared to MiSeq kits, or things like that, highly simplified loading at the start of the run, simplified waste disposal on the cartridge. These really provide a lot of ease of burden to the total cost of ownership on NextSeq and are some of the not most immediate benefits that customers are considering.
So I see one question pop up, which is, can I reanalyze data using onboard DRAGEN? That's such a great question. You cannot actually reanalyze DRAGEN data on board the NextSeq 1K, 2K, but you could purchase or you could get a DRAGEN server or use DRAGEN on cloud, whether it's base-based or ICA, to reanalyze that data. So that's definitely possible. And then there's one more question that I see. What is the lag time between base-based and onboard DRAGEN software updates? So this is also related to another question that I've seen here on this Q&A. Just give me a second. What is on the roadmap for DRAGEN onboard, and will you have a metagenomics pipeline on NextSeq 1K, 2K? So the answer for this, both are quite related. DRAGEN onboard has some of our most popular pipelines, and it is tailored to the needs of our onboard customers. It's quite accessible and easy to use with a graphical user interface and streamlines the data analysis process. Whereas DRAGEN on-premise or DRAGEN on base-based and DRAGEN on ICA, they have some of the most frequent updates. They are highly customizable and offers a comprehensive set of pipelines. To answer both of those questions, so DRAGEN on base-based will always have the most up-to-date pipelines and will have more wider applications when compared to DRAGEN onboard. But having said that, we do continuously, as you've seen, we continuously update the pipelines on board DRAGEN, and we consider user requests like metagenomics, etc., into our planning process. So I hope that answers the questions, both of those questions together. For DRAGEN variant calling, do users upload a reference genome for variant calls, or does DRAGEN just call variants against the consensus de novo assembly from the sequencing run? So users can upload reference genomes. Illumina, we provide references for customers to download, so you can use that to upload, you can either upload those or use your reference genomes.
So it looks like there is another question that says, while we love the higher output offered by the new NextSeq, we also rely on MiSeq V2 500 nano kits for certain projects. Is there any plan to bring longer reads, such as at least 2x250 to the mini-seq, to take the place of this MiSeq niche? This is one of the things preventing our lab from upgrading. That's a good question looking forward. I would say that, there isn't any other plans. I think there was a second question I could sort of address at the same time on plans to get rid of the MiSeq now that we have these new 600 cycle kits on NextSeq. We view the MiSeq and the mini-seq both as sort of this different output range than the NextSeq 1000 and 2000, with the 1000, 2000 sort of having a minimum of 100 million reads, and then the MiSeq and mini-seq having a maximum of 25 million reads, and especially the nano kit, I believe, has only 1 million reads. So those are really outputs that aren't really touched by the NextSeq. So we totally get the difference there, and I don't think we have any plans to discontinue the MiSeq or the mini-seq until we have other options in the output range that they offer right now. So with these new NextSeq kits, these are not going to affect the availability of MiSeq or mini-seq flow cells. And in terms of having that niche, if you're really down there in that sort of like lower throughput area with 1 to 25 million reads, and you don't have projects that could take advantage of the 600 cycle kits on the NextSeq with 100 or 300 million reads, my suggestion would be to stick on those MiSeq nano kits for right now as your best option. I don't think we're bringing any longer reads to the mini-seq anytime soon, but we're definitely looking at new options in the low throughput space. It looks like there is another question on, could you use Nextera XT for library prep? Yes, absolutely. All of our Illumina library preps are totally valid for the NextSeq 1000 and 2000 and all of our instruments. Nextera XT is a totally valid option for library prep, capable with that system. That is actually one of the advantages of just using Illumina library prep with Illumina sequencers. All of our library preps are valid with all of our sequencers. It looks like, the next question, will XLEAP long-read chemistry support RNA applications? So quick clarification, XLEAP, and I think there's another question as well that just asked, what is XLEAP? So that actually might be a good one to address at the same time. So XLEAP SBS is a newer version of our SBS chemistry. We've revamped nearly all of the reagents, chemicals, and sort of players such as polymerases and linkers and dyes, et cetera, things like that. We've revamped nearly everything involved in our chemistry. It's the same process as standard SBS, but due to these improved reagents, it's incredibly more stable, it's faster, and it's able to provide higher quality results. And so what we're doing is we're making all new sequencers going forward with XLEAP SBS chemistry specifically. The first version of that was our NovaSeq X. And we're also bringing XLEAP SBS chemistry in terms of backwards compatibility to one of our existing sequencers, which is this NextSeq 1000 and 2000 product line. So that is essentially what XLEAP chemistry is. It's going to be a revamp of the chemistry that improves on a bunch of different metrics, including price, output, data quality, stability, potentially even read lengths as well. And so when we say XLEAP long read chemistry, these new kits are actually not XLEAP. These are based on the traditional chemistry. So we're first going to have XLEAP higher output chemistry on P4 with the 500 gigabase 500 gigabase kit, and then we're going to revamp the existing kit. So that includes all of the kits P1 to P3, including 100, 200, 300 cycle kits, as well as these new 600 cycle kits as well. They will all support both DNA and RNA applications, depending on how you use them. If you're looking for how long read chemistry can support RNA applications in general, I could definitely say we have new options to support, say, metatranscriptomic reads on this sequencer in terms of, we have a library prep, you can use our Illumina total RNA prep with a new add-on, Ribo 0 plus microbiome, that gets rid of both human and bacterial rRNAs. So you can look at sort of like a total transcriptomic view of a bacterial sample, and that's a really good application for looking at RNA of mixed bacterial samples. I think we have time for one more question.
Does DRAGEN support microbiome analysis of 16S? So it's currently not on board the NextSeq 1000, 2000, but having said that, there is an app for 16S analysis on base-based sequence hub that you can leverage.
All right, I think that's all the time we have for today. Thank you to everyone who was able to join in on today's webinar, and a big thank you to our speakers, Sam and Deepthi.